Chapter 3, Segment 1, pages 59 through 68. Generation, transmission, and integration of neural signals. Neurophysiology is the study of electrical and chemical processes in neurons. Information flows within a neuron via electrical signals, versus information passes between neurons through chemical signals. Action potentials are electrical signals produced by neurons that are then sent along their axons. Neurotransmitters are a chemical messenger that a neuron's electrical signal causes the axon terminal to release, and it goes into the synapse. Electrical signals are the vocabulary of the nervous system. All living cells have an electrical charge that is more negative on the inside than on the outside. Early single-celled organisms contain many proteins that are negatively charged. Early on, neurons began to exploit this electrical property to keep track of information, which resulted in cellular signaling system, which is almost the same in us, jellyfish, insects. These electrical signals underlie the whole range of thought and action, from anything we do, from composing music, feeling an itch on our skin, or swatting a mosquito. A balance of electrochemical forces produces the resting membrane potential of neurons. A neuron at rest, not being disturbed by other neurons, nor is it generating its own signals. An ion is an electrically charged molecule. Of the many ions a neuron contains, the majority are negatively charged anions, especially large protein anions that cannot exit the cell. The rest of the ions a neuron contains are cations, which are positively charged ions. All of these ions, though, regardless if they're negative or positive, are dissolved in cytoplasm, which is an intracellular fluid. A cell membrane, a lipid bilayer, which is two layers of linked fatty molecules that separates the cytoplasm from the extracellular fluid. This fills the spaces between cells in the vascular system. There are more negatively charged molecules inside the neuron as opposed to the outside. A microelectrode measures the difference in charges across the membrane by inserting an electrode into the neuron and using a voltmeter to compare the intracellular and extracellular fluids. These measures show that a neuron has a resting membrane potential of -50 to -80 millivolts. Ion channel, a tube-like pore that allows ions of a specific kind to pass through the membrane. It is an important membrane-spanning protein. The neuron cell membrane comprises of many channels that only allow potassium ions to cross the membrane. Selective permeability. The tendency of a cell membrane to allow some ions to go through or exit the membrane freely, but it inhibits other ions from passing. The channel that allows only potassium ions to cross the membrane has selective permeability to potassium ions. The balancing of two opposing forces that drive potassium ions in and out of the neuron is the resting potential of a neuron. Diffusion. A force that causes certain molecules of a substance to diffuse from regions of high concentration to low concentrations. Molecules will move down their concentration gradient until there is an even dispersion of concentration in that location. When a membrane divides two areas, the potassium ions will flow through the membrane until there is an even concentration on both sides of the membrane. If a particular substance cannot cross the membrane, it stays concentrated on the one side. Sodium-potassium pump, a mechanism utilized by neurons that pumps 3 sodium ions out for every 2 potassium ions pumped in. This causes a buildup of potassium ions inside the cell, but because the membrane is permeable to potassium, these ions will down their concentration gradient, resulting in a net buildup of negative charges inside the cell. A negative charge accumulation results in the cell exerting electrostatic pressure to draw positively charged ions back in. The concentration gradient pushing the potassium ions out of and the electrostatic pressure pulling the potassium ions in will eventually reach an equilibrium, so that any further movement of ions into the cell is matched by the flowing of ions out of the cell. Equilibrium potential is the voltage difference across a permeable membrane needed to counterbalance the diffusion force. We can calculate this with two equations. The Nernst equation, which predicts the equilibrium potential for a given ion based on the concentrations of the ion on opposing sides of a permeable membrane. The Goldman equation, on the other hand, predicts the potential difference across the membrane based on the concentrations of ions on opposing sides of the membrane, as well as its relative permeability to each ion. The Goldman equation is closer estimate to the resting potential observed in neurons because it, first of all, takes into account the extracellular and intracellular concentrations of sodium and potassium, as well as several other ions, and it accounts for the degree of permeability to each. Another reason why the Nernst equation isn't as accurate as the Goldman equation is that it assumes that the neuronal membrane is only permeable to potassium ions. The membrane is semi-permeable to a few other ions as well. The resting potential of a neuron is a foundational level of polarization found in all cells. Unlike other cells, neurons continuously experience a short-lived yet extreme change in polarization. A threshold amount of depolarization triggers an action potential. Now we dig deeper into our action potential definition. It is a brief but large change in a neuronal polarization that are transmitted along the axon at high speeds. Information that a neuron sends to its postsynaptic targets is encoded in patterns of action potentials. There are two important concepts as to how action potentials are triggered. And these are, first of all, hyperpolarization, which causes the inside of the neuron to become even more negative than the outside of the neuron. A resting potential could be negative 65 millivolts, but with hyperpolarization, the resting membrane potential may become negative 70 millivolts. This causes the resting membrane potential to travel even farther away from zero, whereas depolarization decreases polarization of the cell membrane, which brings the membrane potential closer to zero. The outside of the neuron becomes more like the inside of the neuron. A graded response is a neuron's change in potential, which is applying a hyperpolarizing stimulus to a membrane, which causes a swift response following the stimulus pulse. The greater the stimulus, the greater the response that will occur. Local potentials. As you get farther away from the stimulus, the membrane's potential decreases. It's basically a physics law. As the potential spreads across the membrane, its size decays as the function of a square of the distance. Graded and diminished over time in distance will also arise at the synapse in response to other neurons. Application of a depolarizing stimulus will produce the same reaction as hyperpolarizing the stimulus to a certain extent. If the stimulus depolarizes the cell to its threshold, which is negative 40 millivolts, an action potential, or spike, is provoked. An action potential is an instant reversal of the membrane potential that momentarily makes the inside of the membrane positive with regards to the outside. The action potential is actively regenerated down the axon throughout ionic membranes. When you apply a strong stimulus that exceeds the neuron threshold, a large depolarization, you will see more action potentials. This brings us to our all or none property. An action potential will either fire at its full amplitude or will not fire at all. Information is encoded by changes in the rate of action potentials and not in their size. An active potential is the positive or negative change in membrane potential that may allow an action potential. The return to baseline membrane potential is complex. Many electrons will undergo electrical oscillations immediately after their action potential.